Hello everyone, and thank you for attending today's webinar, The Brain on Drugs, the Strategic Use of Cognitive Measures and Biomarkers in Early Phase CNS-Drug Studies. I'm Sharon Salt, Senior Editor of Neurocentral, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. All the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. A copy of today's slide deck and our feedback survey are located in the resource list. We encourage you to download any resources or links that you may find useful. Please also let us know your thoughts on today's webinar by tweeting us at neuro_central or by using the hashtag #NCWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks can cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we are delighted to have both Denise Milovan and Beatrice Setnik from Altasciences presenting for us today. Dr. Milovan joined Altasciences in 2020. In previous neuroscientist roles at INC Research / Syneos Health and Decision Line Clinical Research, she provided expertise and oversight of neurocognitive early phase programs dedicated to the assessment of the pharmacodynamic effects of CNS drugs. Dr. Milovan practices clinical neuropsychology in a variety of settings, including hospitals, nonprofit organizations, and private practice. She is currently the Vice President of the Council of the College of Psychologists of Ontario. Dr. Milovan holds a PhD and master's degree in clinical neuropsychology. Dr. Setnik has been working in the area of clinical drug development and abuse potential assessment since 2005. Dr. Setnik is currently the Chief Scientific Officer at Altasciences and an adjunct professor at the University of Toronto in the Department of Pharmacology and Toxicology. Dr. Setnik earned her doctorate degree in pharmacology and the collaborative program in neuroscience from the University of Toronto in 2005. She has published numerous research articles in internationally recognized peer-reviewed journals and is a recognized expert in the fields of human abuse and physical dependence potential evaluation. We invite the audience to submit any questions you may have for our presenters throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Denise and Beatrice in a live Q&A session after the presentation and then follow up any outstanding questions offline. Thank you again for joining us today, everyone. I'll now hand over to Denise and Beatrice to start today's presentation.
Thank you, Sharon, for the very nice introduction. Today's webinar is Brain on Drugs, Strategic Use of Cognitive Measures and Biomarkers in Early Phase CNS Drug Studies. Dr. Beatrice Setnik, Chief Scientific Officer at Altasciences, and myself, Denise Milovan, Neuroscientist and Scientific Manager at Altasciences, will be your presenters today. This webinar will highlight potential advantages of using targeted cognitive markers during clinical drug development. The relevance of cognitive endpoints to guide clinical trial design will be addressed. Dr. Setnik will present cognitive test choices and will rely on a practical example to illustrate the advantages of incorporating cognitive tasks for go-no-go decisions. She will discuss the benefits of including biomarkers in early phase studies. The field of neuropsychology began as the attempt to understand how brain lesions change our ability to carry out our day-to-day activities. We now define neuropsychology as the study of the relationship between nervous system structures, the study of the brain, and their corresponding behavioral and cognitive functions, the study of the mind. At the beginning, and in the absence of any specific knowledge as to what areas of brain activity were measured by any particular test, Neuropsychologists have relied on the comparison between one individual's scores against those from a larger group of their same age, same-sex peers. They used statistical analysis to determine if any of these differences in performance were meaningful. Over time, the use of cognitive tests in individuals with brain lesions has made it possible to identify cognitive domains that, broadly speaking, reflect specific brain behavioral relationships. And although medications are routinely used to manage the behavioral consequences of brain lesions, the cognitive effects of medications have received comparatively little attention both in research and in clinical practice. The latest version of the Diagnostic and Statistical Manual of Mental Disorders, DSM-5, identifies 6 major cognitive domains that span complex attention, executive function, learning and memory, language, social cognition, and perceptual motor function. Of all the cognitive domains, attention is central to the study of neurocognition. Basic attention is seen as the prerequisite for all mental functions, and is understood to represent the capacity to sustain our focus long enough to complete a simple task. However, in order to understand complex cognitive processes, distinct attention mechanisms have been grouped together, and they have been defined as the ability to control, to shift, and to divide our attention in order to manipulate information and to execute the multitude of steps that are generally necessary to accomplish goals. The complex attention domain can be subcategorized into the ability to sustain attention or to stay vigilant, divide our attention, which is generally seen as multitasking, focus attention, or engage in selective processing of information, and also the ability to process information rapidly and accurately. Each of the attention subcategories can play a critical role in our ability to engage and understand the world. The executive function domain reflects cognitive control and involves processes required for goal-directed behaviors. It encompasses planning, involved in motor planning and programming, working memory, or the active use of information until completion of an action is possible, inhibition, or the control of automatic reactions to encourage adaptive behaviors to take place, flexibility, the ability to constantly adapt to changing environments by shifting between different mental sets, tasks, or strategies, problem solving, or the ability to deal effectively with complex, non-routine situations across all the different cognitive domains, and decision-making, or the complex coordination of several cognitive processes that usually take place at the same time. Both distinct and intertwined, the learning and memory components of the learning and memory domain combine to form short-term memories, defined as the capacity to keep in mind a small amount of information in a readily available state for a short period, while long-term memory proceeds through 3 distinct phases of encoding, which is the initial registration and acquisition of information, storage, or the maintenance of information over time in the nervous system, and retrieval, the process whereby the stored information is brought back into conscious awareness and is otherwise used to affect ongoing behavior. The language domain refers to the ability to produce and comprehend both spoken and written words. It can be categorized as expressive language, which includes naming, word finding, fluency, grammar, syntax, and receptive language, which is concerned with reading and listening. The social cognition domain focuses on the role of cognitive processes in social interactions. It is the study of how people process, store and apply information about others and about social situations. As such, it relies on recognition of emotions, having a theory of mind, or being able to attribute and understand our own mental states as well as those of others, and finally, on having insight, or the ability to evaluate and reevaluate our thoughts and beliefs in order to reach thoughtful conclusions. Perceptual and motor functions comprise the sixth and final cognitive domain. This domain covers fine and gross motor skills, motor coordination, integration of visual and motor skills, integration of the different senses, as well as tactile perception. Neuropsychological testing is concerned with how insults to the brain impact various aspects of cognitive domains. To this effect, modern neuropsychology relies on non-invasive tests to inform on the presence of brain damage, to hypothesize about the localization and lateralization of the damage, draw conclusions regarding the etiology of the impairment, and offer prognosis for recovery. Comprehensive testing batteries are generally administered in one-on-one interactions, either via pen and paper or computer-based assessments, or even a combination of the two. These batteries allow clinicians to gather converging data to make final diagnoses and to provide detailed recommendations for cognitive remediation. However, given our increased understanding of medication-induced neurocognitive effects, clinical recommendations are now more and more used to also guide the selection of pharmacotherapies that aim to minimize potential negative cognitive effects. For example, continued use of anticholinergic medications in individuals with already reduced central cholinergic activity, such as those with mild cognitive impairments, can increase the risk for cognitive complications. In rare examples, such individuals may even experience delirium. Antihypertensive medications such as propranolol, a beta blocker, and methyldopa have been shown to correlate with increased verbal memory deficits, and beta blockers have also been associated with heightened risk for drug-induced depression. The use of such medication, then, may not be advised for either depressed individuals or those with memory deficits. Pharmacologic research into the major CNS drug categories, stimulants, depressants, hallucinogens, has primarily focused on the psychological effects of the drugs investigated. In human abuse potential studies, for example, inferences regarding risk for abuse rely heavily on the presence of desirable psychological or subjective effects, such as euphoria, hallucinations, and other perceptual distortions or changes in mood. Although alterations in cognition can and do impact on the likelihood that the drug may or may not be abused, objective measures have remained a secondary focus in these trials. FDA noted that the occurrence of adverse CNS events in even a small number of phase 1 subjects can indicate the need for more focused studies of CNS effects. It is then easy to see how the careful evaluation of potential cognitive safety effects can help identify whether an investigational product may either ameliorate or further impair the cognitive profile of patients or subjects. The question then becomes whether cognitive safety measures should be routinely adopted into early phase studies that normally concern themselves with issues of safety, tolerability, and dose selection. Should cognitive safety become an integral part of early clinical trials? Careful consideration should be given to selecting appropriate measures that correlate to the cognitive domains potentially impacted by the investigational product. Incorporating cognitive safety into enhanced early phase trials can help improve the identification of dose-response relationships, improve the selection of safe doses for later phase development, can help establish the cognitive safety profile of the investigational product as compared to that of competitor drugs, can assist in evaluating the risk-benefit ratio for emergent undesirable cognitive effects, can help clarify whether additional evaluations during later phase trials may be needed or may even be waived, and can help identify early on if post-marketed cognitive function monitoring may be required. If we decide to include cognitive measures, then what should our number one selection criterion be? FDA noted that early testing for CNS impairing effects should generally emphasize sensitivity over specificity. One illustrative example is that of the DSST, digit symbol substitution test, is one of the most commonly used neuropsychological tests, and it has been shown to demonstrate high sensitivity to change in the presence of cognitive impairment or improvement in both acute and chronically ill patients and across a wide variety of clinical populations. In addition, it is fast to administer. It takes somewhere around 1.5 minutes to 2 minutes. It is reliable, and it is minimally impacted by language. It can be administered via pen and paper or using computer administrated testing. Its drawback is low specificity as to which cognitive domain may have suffered the impairment. However, in clinical practice, this type of measure has been found to be very useful at the level of individual patients for making medication and dosing decisions. Similar benefits can then easily be anticipated when selecting highly sensitive measures to assess the cognitive profile of investigational products. The field of neuropsychology uses a wide variety of measures that can provide meaningful information about all the cognitive domains. Thus, depending on our knowledge of the expected impact of the investigational drug on cognition, sensitive, reliable, and informative testing batteries can be selected. Here are a couple of examples of CNS drugs and cognitive interference. The trace reaction time test is highly sensitive to the impairing effects of CNS depressant drugs, such as alprazolam. The two graphs depicted on this slide show CRT's ability to differentiate not only between the effects of alprazolam compared to placebo, but also between the two different doses of the drug. This slide captures the linear impairing effects on response accuracy and slowed responses over time for the highest versus lower alprazolam dose. The magnitude of cognitive effects can be used to make comparative inferences about the relative impact of various doses of agents such as alcohol, midazolam, propofol, or fentanyl on performance. DSST performance was markedly reduced during alcohol intoxication, blood alcohol concentration of 0.11%, with scores approximately 10 points lower than those of placebo. In addition, DSST performance over time was significantly different following administration of midazolam compared to placebo, but not when compared to the effects of alcohol. And here are a couple of examples of CNS drugs and cognitive enhancement. In a double-blind, placebo-controlled, within-subject design study, 15 subjects received an acute administration of cocaine, 300 milligrams, and cannabis, 450 micrograms per kilogram. Cocaine was associated with improved divided attention performance, manifesting as fewer tracking errors and increased response accuracy relative to cannabis. In the same study, cocaine administration was associated with improved performance on a prospective memory task relative to both placebo and cannabis. And now I will hand the presentation over to Dr. Setnik, who will continue to address cognitive test selection in early phase trials.
Thank you, Denise. Now we'll turn the presentation over and discuss the cognitive test selection for early phase clinical trials. There are various opportunities throughout the early phase drug development process to strategically include a variety of cognitive tests to evaluate CNS active drugs. This can begin with preclinical data, observing such things as excitability or possible CNS depressant effects by observing animal behaviors. Moving on to clinical trials, one can use a variety of different cognitive tests and batteries that span various aspects of cognition, including memory, sustained attention, psychomotor speed, amongst others. These types of cognitive measures can be incorporated into a variety of early phase trials as early as your first-in-human single ascending and multiple ascending dose trials. The cognitive results of these trials are very useful in informing future direction of your drug development pathway, determining if there is evidence of impairment or enhancing effects of your CNS active drug, the potential need for additional studies to satisfy regulatory requirements, and determining if such studies are needed, what kind of active controls would be most relevant based on the pharmacology of your drug. There are various important aspects to consider when including cognitive testing in your early phase trials, various do's and don'ts to consider. You want to be able to use these types of sensitive tests in a sparing fashion in early phase research, tests that are most relevant to the pharmacology and the anticipated mechanism of action of the drug. These tests can provide very informative data for later studies. However, they must be carefully selected to ensure that you have information that you need to make critical drug development decisions later in the pathway. You also want to consider using tests that can bridge data from the animal models from your preclinical studies over to those clinical trials in terms of translational research. And consideration of sample size and power is important, particularly if you're designating these kinds of cognitive tests as primary or secondary measures in your trials. Some of the things you'd like to avoid are the use of sensitive scales in the wrong populations. For example, the drug liking visual analog scale is validated and used routinely for recreational drug users. However, its usage in healthy volunteers can lead to misconception of the meaning of the terms as well as some erratic data. You also want to be able to limit the testing to those that are most relevant for your drug. Using every type of testing may create burden in a clinical trial, which leads us to the next point of over-administration. Doing very frequent testing can lead to subject fatigue, which may decline in performance simply as a function of repeated and prolonged administration as opposed to a drug effect. Another important element here is to have both subjects and staff properly trained to administer any such cognitive measures. To illustrate an example of how useful early cognitive testing can be for a clinical drug development program, I will now present a case study. Our case study for today is flibanserin. This is an unscheduled drug indicated for generalized hypoactive sexual desire disorder. This drug is centrally active and causes CNS depression with adverse events such as somnolence and sedation. You'll also note that these effects are quite common for other CNS drugs such as opioids as well as benzodiazepines. In the development program for flibanserin, various cognitive studies and evaluations were included, including studies such as the alcohol interaction and the driving simulator study. Data from cognitive testing is informative in drug labels. For example, for flibanserin, under warnings and precautions, The study data related to the interaction with alcohol has been summarized in section 5.1. This is important for patients who may be taking the drug and have an understanding of what type of interactive effects may occur if the drug is taken concomitantly with alcohol. The effects of driving were also summarized in the flibanserin label in section 14.2. This label shows the results of a driving simulator study that showed that there were no adverse effects detected on the driving performance. As you can see from the figure, the positive control zolpidem showed more impairment effects on driving simulation compared to the 2 doses of flibanserin studied. Through the drug development program, the FDA and in turn the DEA gave flibanserin an unscheduled status in that there was no signals for euphoria and signals indicative of abuse potential. This was made possible because of the extent of cognitive measures and pharmacodynamic testing that was conducted throughout the trial to show that there were no concerns related to abuse or dependence potential. This is an important strategy for sponsors, particularly when you have drugs acting centrally and in being able to evaluate whether or not additional scheduling controls are needed for your drug. Now we'll turn our attention to biomarkers in neurology. Biomarkers are useful indicators that can be objectively measured and can be an indicator of physiological or pathological processes, for example, for diagnostic purposes, or they can also be very useful for determining pharmacological responses to a therapeutic intervention or drug. They are very useful for looking at purported mechanisms of action, possible toxicity, and evaluating the pharmacokinetic profile of a drug and whether a drug induces or inhibits enzymatic activity. Neurological biomarkers are typically present in the CSF, but some have been coming to light that they can be usefully and easily accessible by plasma concentrations. The research, however, is still ongoing in this space, but this offers a very exciting opportunity to look at and diagnose and determine drug effects for various conditions in both neurology and psychiatry. At Altasciences, we have several biomarkers for neurology that have been validated with assays. These are different types of markers that are associated with different types of indications, including traumatic brain injury, Parkinson's disease, and Alzheimer's disease, to name a few. But here are a few examples of biomarkers that have been used and appear in drug labeling information. Our first example is Tegretol, which is carbamazepine. These are extended release tablets. You'll find that in the warnings section of the label, there is a note of serious dermatological reactions associated with patients carrying the HLA-B1502 allele. This poses a safety risk, and being able to assess this important biomarker helps clinicians mitigate this risk in the patient population. In this next example, the biomarker is used as a diagnostic tool. Exondys 51 is indicated for patients who have confirmed mutations of the DMD gene that's amenable to exon 51 skipping. By using this important biomarker to identify the appropriate population enables clinicians to identify correctly which populations have this disorder and should be treated with this particular drug. In summary, there are many benefits to including cognitive testing as well as potential biomarkers in early drug development. This helps inform the safety profile of a drug, the risk of impairing effects and other effects that may pose safety concerns later in clinical drug development, as well as the eventual marketing of the drug in the real world. Cognitive testing also enables us to evaluate important pharmacodynamic drug effects and characterize the pharmacology of a drug early in drug development. And it also can be used to support proof of concept to illustrate mechanism of action and determine if a drug exerts certain effects that it's expected to. Early development and early inclusion of cognitive testing also may serve to waive additional studies that may be dedicated studies needed later on, such as abuse potential or driving simulation, or may warrant and signal the need for those studies and help you plan with enough data collected early on to design more informative studies later in drug development. So there are a number of really good reasons why cognitive testing and biomarkers are very useful tools in early drug development. Thank you for listening. We'll go on to questions.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you do have any questions for Denise or Beatrice, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We've received a number of questions so far, so we'll now address these live on air. So our first question is, data from animal studies suggests that cognitive effects may be present and such measures are planned for late-stage studies. Why would inclusion of cognitive tests early on provide added value?
So I can take this one on initially to start. The early measures of cognition give you a very early read-in whether or not you have impairing effects of a drug. And so there are important safety considerations to assess from the onset in early drug development so that you can better inform your drug development program as to whether or not additional further dedicated studies may be needed for impairing effects, for example, driving simulator studies, or if those could be waived if you don't have if you have an absence of signal. You may also be looking at these types of studies to determine if there may be required abuse potential testing, if there are some indications that there are adverse events or certain measures that may be being endorsed early in clinical trials. But also the other component, in addition to safety, is also the initial efficacy. So if you're developing a drug that may have cognitive enhancing effects, including some of those measures early into development can give you some read-in into the proof of concept of the drug or an endorsement of the mechanism of action that is doing the same intent in humans. So there are a number of reasons why this allows you to have better insight into then powering later further testing that's done later in clinical trials, inclusion of important safety parameters and considerations for later patient trials, as well as the regulatory strategy as to whether or not you will need to conduct certain dedicated studies or have the potential to waive those studies in your development program. So many advantages to consider.
And in addition, if we are to think about a very practical example regarding the transition from animal studies to human studies, for example, cocaine exposure in rats has shown that drug addiction can have detrimental effects on the ability of the animals to navigate through space and to learn and to remember information. And if we are considering a bridging of the data from the animal studies into human studies, we might consider a measure such as parent-associated learning that would provide us additional details about the ability of the test drug to impact on learning and memory in adults.
Thank you both. So the next question that's now come in says, are there any guiding principles in selecting which are the best cognitive measures to include in a study?
So I'll take this one on. There are really two main areas for measuring cognition. One is to evaluate the safety of a drug and its CNS activity, and the other one is related to its activity and efficacy and mechanism of action. So if we take the first one related to safety, when we're designing trials as early as our first in human studies, we're really designing those trials to look at safety at a wide range of doses under both single and multiple dose administrations. This is an optimal time to determine if a drug causes any degree of impairing effects that could pose safety issues not only in a patient population, but in later clinical trials. The absence of such signals could also serve as relevant waivers in your regulatory navigation of your drug development program to potentially waive dedicated studies related to impairment, such as the driving simulator study. So these become very important safety evaluations that are best conducted. And the types of measurements that you would look at from that effect, if you're looking at an impairing effect, could be things like the psychomotor speed, reaction times, the ability to have sustained attention, things that you think about in terms of being able to operate a complex machinery like a car, for example, some of these surrogate measures. On the flip side, there are a variety of different things that CNS drugs do. They're not only do they cause impairing effects, they can cause enhancing effects that could be related to the efficacy of a drug, improved memory, improved attention, as you see in some disorders as ADHD. So in these types of cases, you may also be looking at the flip side of the CNS activity and how it may relate to the efficacy. And the early types of studies may have some limitations because you're looking primarily at healthy volunteer models. However, there are possibilities of including smaller subsets of patients into these trials, and some measures may be sensitive enough and population sensitive enough where you may be able to detect changes in the measures of cognition, even in healthy volunteer populations, as preliminary kind of confirmations of the mechanism of action. And I'll hand it over to Denise to further elaborate on some guiding principles for selecting these measures.
And choosing sensitive measures early on in clinical trials may also inform us to the recovery profile over time. And this information is actually going to be quite useful in terms of dose selection and choices of measures that can be used later on in studies that are going to include the targeted patient population.
Perfect. Thank you both. So the next question asks, what can data from cognitive tests given to healthy volunteers tell us about a drug expected to prevent cognitive deterioration in patients with neurodegenerative disorders such as Alzheimer's disease or Parkinson's disorder?
Thank you for this question. I will take this on. The patients who are diagnosed with Alzheimer's disease or Parkinson's disease really do get diagnosed relatively late in the disease progression, and even when they get diagnosed earlier in the disease progression, oftentimes this happens because they are experiencing deterioration in their cognitive performance. And then it is a very fair question to ask ourselves, how can data from healthy volunteers who are not experiencing any cognitive deficits be informative with regards to neurodegenerative disorders and cognitive impairment? And one of the ways in which we can address this question is by using a scopolamine model, for example. Scopolamine is a drug that induces reversible cognitive impairments, and as such is very well suited to allow researchers early on to set up a strategy to examine very specific cognitive deterioration that can be reversed, and to do so safely before asking patients to be exposed to a test drug.
Great, thank you, Denise. So the next question that's now come in says, psychological subjective effects like drug liking, good drug effects, and the desire to take the drug again are considered to be determinant factors for drug schedule classification. What role, if any, can objective cognitive tests play in scheduling decisions?
It is true. Psychosocial factors are certainly critical to understanding the potential for abuse. However, cognitive factors such as enhanced attention capability, for example, can lead to improved performance in work, in school, or perceptual changes that are associated with some of the drugs can actually augment creativity and be perceived as enhancing cognitive performance. And so understanding the cognitive impact of a drug may be quite beneficial with regards to scheduling decisions. For two drugs that are in the exact same class of drug, the difference between whether cognitive effects are present or absent may be the difference between one drug being abused and one drug not being abused, hence not being scheduled.
That's a great answer. Thank you very much. So next up we have, how can sponsors be confident that subjects are going to perform well on the measures?
Well, there are a few things to consider for performance on measures. And the primary consideration is ensuring that both your staff and your subjects are well trained. The staff need to be able to be very well trained on the administration measures. The environment needs to be correct and non-distracting for subjects when they're answering these questions. And secondly, the subjects need to understand the measures, how to use the interfaces on which the measures are being administered, and have comprehension of the questions of what's being asked of them. This will help ensure that the best appropriate answering and conduct of these measures are implemented. The other component of this is when you have measures of cognition or things that may have a learning curve for them, there needs to be an adequate time to train subjects and give them enough time to practice on those measures so that they can overcome the learning curve. Otherwise, if this isn't done, this could falsely look like a drug effect when it's actually a learning curve. So the administration is very important, and appropriate administration is important as well. The other element of this is also ensuring that when you administer measures throughout your clinical trial and on any given dosing day, you're not overburdening the amount of measures. If the measures are very long and frequent, subjects will have fatigue and burnout, and the performance will deteriorate as a function simply of overzealous administration of tests rather than a drug effect itself. So one does need to be very careful in balancing what is an appropriate sampling time for a drug effect, and note which time points are the most appropriate for the time course of the drug. And lastly, part of the success of this is also making sure that your measures are validated for the populations that you're looking at. When the measures are not valid or not appropriate for a population, you will have erratic responding that just simply doesn't make any sense. So all of these factors combined help ensure successful implementation of cognitive and other subjective drug effect measures into your clinical trials.
Thank you very much to both Denise and Beatrice for answering our audience questions today. So before we wrap up, do you have any last comments that you would like to add?
Yes, first of all, I wanted to thank everyone for joining today and for your interest in this topic. The takeaway here is that inclusion of cognitive measures and pharmacodynamic measures early in your development program can really help set the stage for both a very effective drug development program as well as a very good regulatory strategy in understanding the limitations and the benefits of a product. Certainly, the correct inclusion and the best application of cognitive measures really needs to rely on working with a CRO and a site that has a lot of experience running these measures, selecting measures, and administering these measures, and including the training of staff and subjects. Denise and I are both experts in this area. We work with very highly trained clinic operation folks and investigators who routinely include cognitive measures in early phase trials. And should there be any questions that you would like to reach out to us directly, we are more than happy to help answer and help guide you on your specific development program. So thank you again for joining.
And certainly choosing cognitive measures might sometimes feel daunting. However, combining our knowledge with the data from preclinical studies and the expectations of what the impact of a drug might be on cognition will help navigate these difficult decisions to arrive at a right choice for any drug of interest.
Brilliant. So I want to once again thank our wonderful presenters, Denise Milovan and Beatrice Setnik, as well as you, our listeners, for your time and questions today. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at neuro_central. For more webinars, please visit NeuroCentral at www.neuro-central.com/webinars. So these next two slides include references from the webinar, so if you are interested, then please do take a look at them. Thank you again for attending, everyone, and goodbye.